Hi everyone. First, I would like to thank the organizing committee for giving me this opportunity to talk to you about using non-clinical comparative methods to evaluate immunogenicity risk of complex peptide products. Before I begin, I would like to make a disclaimer that this presentation reflects my view and not the agency's. In this talk, I'll be first going over the guidance for assessing the generic peptide of synthetic origin referencing peptide of recombinant origin, especially focused on the immunogenicity risk assessment. And then for the purpose, I'll discuss some examples of in silico and in vitro assays of assessing adaptive immune response as well as an innate immune response. And lastly, I'll mention a few common deficiencies and issues found during the validation and development of these in vitro assays. As you have heard in the previous presentation, peptides can be made through either recombinant process on the left or synthetic process as is depicted on the right. In the recombinant process, some of the process-related impurities such as whole cell proteins can be introduced into the drug substance. Similar with synthetic process, some of the process-related impurities can also be introduced. Although many of these impurities can be removed during the final stages of API purification, the removal is never 100%, and so many of these impurities can end up in the final drug substance and further into the drug product. And it's for these reasons that we are really concerned about some of these impurities may cause risk for immunogenicity. So what is immunogenicity and why do we care about it? From our experience with protein products, we know that immunogenicity may impact the safety and efficacy of the drug product. In some cases, the development of anti-drug antibodies, such as neutralizing ADAs, can affect the PK and diminishing the drug effect. In other cases, the anti-drug antibody can cross-react with endogenous non-redundant proteins in patients, and this may cause deficiency syndromes. In rare but severe cases, the hypersensitivity in the form of cytokine release syndrome and anaphylaxis can also cause serious consequences. Therefore, when it comes to seeking ANDA approval that are synthetic peptides referencing recombinant RLDs for these longer peptides, we have provided some recommendations on assessing the immunogenicity risk of the proposed generics in this guidance. This guidance applies to glucagon, liraglutide, nesiritide, teriparatide, and tetraglutide. Next, I would like to go over some recommendations pertaining to the peptide-related impurities. First of all, the impurities found in both generic and RLD products, these common impurities in the generic should be less than in RLD. For any new impurities that are found only in the generics and not in the RLD, we will refer to these as new impurities, and the acceptable limit for these should be no more than 0.5%. And the reason we believe in most cases, these synthetic related impurities can be controlled down to a reasonable level. Because we want to limit the potential risk of immunogenicity from these impurities coming from the synthetic process, we are recommending that the ANDA applicants to seek submission for highly purified peptide product, as outlined in the guidance. The new impurities in the range of 0.1 to 0.5%, we recommend these to be identified, characterized, and justified for not affecting the safety efficacy of the product. And for this part, the assessment should also include immunogenicity risk assessment, which next I'll be discussing some non-clinical immunogenicity risk assessment assays. So first, I would like to make a brief introduction to innate and adaptive immunities, as the guidance made reference to both of these immune systems. On the left depicts the cells involved in the innate immune response. The innate immunity are typically the first responders to infections and fend off foreign invaders in our bodies. And in the drug product, some of the process-related impurities, such as contaminants and also some extractable leachables, can trigger this response. And in the guidance, the agency recommend that the testing on the whole drug product for the innate immune response should be done. And this recommendation is independent of
whether there are new impurities found in the proposed drug product. And here on the right are some of the key players for adaptive immune system. The lymphocyte, which is boxed in the red, also known as the white blood cells, include B cells and T cells. The B cells secrete the antibodies, and T cells can activate by MHC binding peptide antigens. So their roles include direct killing of the host cells, activate immune cells, produce cytokines, and regulate immune responses. So therefore, any adaptive immune response would depend upon the T cell binding to the peptide that is presented to the MHC. So for any new peptide impurities in the range of 0.1 to 0.5%, we like the applicant to investigate whether these impurities contain an MHC binding domain. Next, I'll give an example of a study where we have done using in silico assessment for determining the immunogenicity risk of a synthetic peptide, and we use synthetic salmon calcitonin. But keep in mind that since salmon calcitonin is not one of the 5 peptides outlined in the guidance, however, it is commercially available. That's why we're using it for this study. Here we have analyzed marketed synthetic salmon calcitonin generics and found multiple impurities across various products. Some of these are related to API stability and degradation, such as oxidation and deamidation. We also found some impurities that are related to peptide synthesis, such as insertions and substitutions. So when we take these synthetic related impurities and plug them into the in silico immunogenicity analysis, what we found is some of these impurities have a higher immunogenicity potential than the API peptide, which is represented here. The API is bolded as labeled RLD, SCT, and with a score of 4.46. And that is the score, potential immunogenicity score. And anything, all these impurities above those would potentially have a higher immunogenicity risk as they have a higher affinity for MHC binding. But if you look on the brighter side, that is, all these impurities, including the highest predicted risk for immunogenicity, are in general lower than most of these immunogenic proteins, including tetanus toxin and influenza hemagglutinin. Another piece of information that we can get from the in silico immunogenicity risk assessment is that when we utilize, this is done on utilizing Epivax's immunoinformatics system to see how each of these impurities can potentially affect the populations of various populations carrying HLA supertype alleles. So in this example, a single amino acid change at protein 23 can cause notable increase in MHC affinity for population or subpopulation carrying DRB1, 4, 11, 15 supertypes, and these are boxed in red at the bottom. And also slight elevation in affinity in DRB7 and 8. As you can see, in silico analysis has its advantage in that it can quickly assess an impurity's immunogenicity potential on a diverse range of population carrying various HLA alleles. However, in silico model and results need to be validated. And to do so, the in vitro assessment can be used. After we conduct the in silico assessment on the salmon calcitonin impurities, we select some of these impurities to do some in vitro assays to validate these risks. And from what we found is that typically the results obtained from the in silico analysis matches those from the in vitro assay, as you can see from this table. However, in some cases, such as this threonine 21 here, the in silico risk assessment overpredicts the risk, which is why it is always worthwhile to verify these results from the in silico analysis using in vitro assays. So in our study, we used 2 in vitro assays to validate the in silico results. These are MHC binding assay and in vitro T cell assays. Next, I'll spend a little bit of time to describe these assays. The MHC binding assay is used to measure how well peptides are predicted to bind to MHC the peptides, or these are fragments of the peptide, are synthesized and assayed over a range of various concentrations in HLA binding assay. The peptides are incubated overnight with soluble HLA, a biotin labeled competitor of moderate affinity. Then the reaction is halted and mixture is transferred to a plate coated with anti-HLA antibody. And then the plates are developed next day, and adding streptavidin, europium, and fluorescent is measured. If you want to learn more about this assay, I refer you to this paper by Java and company. Next is the in vitro T-cell assay. These are PBMC-based assays. The PBMC cells collected from donors are repeatedly added with repeatedly stimulated with IL-2 and IL-7, and also the peptide of interest, in this case the peptide impurity, are added for additional stimulation. The anti-human interferon gamma ISPA is performed using commercial kit, and then the cells from stimulation or later added to the ELISpot plate and read for interferon gamma secreting T cell response. So if you want to know more about how to optimize and run this assay, I will refer you to the paper by Warner and al. So when we are reviewing these assays, we see some common deficiencies being issued to most applicants. First of all, we recommend that the PBMC are from donor population that is reasonably diverse and representative. This may mean to select some PBMC from donors carrying less popular HLA supertypes and more diverse HLA supertypes. Also, the number of donors should be sufficient in size. In order for us to determine if the assay is adequate and fit for purpose, we like to know the details of how the experiments are designed. Therefore, the experimental details such as duration of the assay, number of the cells per well, these should be provided. Next, the sensitivity of the assay should need to be demonstrated. This can be done using peptides at various concentrations to show what is the optimal loading amount of the peptide, test peptides to use. Of course, it is also necessary to provide information and justification on how to determine the threshold for setting various levels of risk. So next, I'll spend some time on innate immune response assays. So briefly, we already previously mentioned that innate immune response are like the first responders when it comes to foreign antigens. And these TLR or toll-like receptors on various immune cells, such as monocyte macrophages, can recognize these inorganic or microbial antigens. So in the drug product, these antigen impurities can come from various sources, such as manufacturing process, shuttle leachables, and so forth. And when the receptors recognize these antigens, it can lead to the activation of the immune system and may further, it can lead to local inflammation, also facilitate the antigen-specific immune response to exogenous proteins, or even help to break the tolerance to endogenous peptide proteins. So when it comes to in vitro assays for assessing innate immune response, here are some of the innate immune cells one can use. And most of these cell lines are human origin with the exception of raw blue, and all of them are commercially available. So PBMC can also be used in the innate immune assays. So PBMC from the donors or whole blood from the donors can be incubated with the drug product, and then cytokines, various cytokines can be measured using commercial kit. The advantage of using PBMC is it may be clinically relevant. You're using human blood. However, donor to donor, there's going to be donor to donor variability, and you need to be aware that preparing, handling, storing this PBMC can be complex and challenging, and also PBMC may lack some of the underrepresented cells such as macrophages, dendritic cells, polymorphonuclear cells. So be prepared to justify this. So instead of working with human whole bloods, one may choose to work with cell lines with specific TRL receptors, such as what's outlined in this table. The advantage of using cell line is it has less variability in compared to PBMC, so it can be very reproducible. It makes it easy to validate and transfer. In addition, it has good sensitivity, as you can see from the study by Hay and al. From Daniela Verthides' lab in FDA. So I'm not going to go into details about how these assays are run, and I'll refer you to this paper here. Next, I'll discuss a little bit about some of the common deficiencies we have cited about the innate immune response assays. One common question we tend to get on confusion from many applicants is, if their proposed generic product has no impurities, can the innate immune response assessment be skipped? And the answer is no, you still have to do the innate immune response. As discussed previously, the purpose is to assess the drug product as a whole, free of contaminants and other process-related impurities. Another issue when reviewing these assays, we find oftentimes there's no clear rationale as to when certain cytokine signals readouts are selected. We think it will be best that the applicant provide some justification or rationale for selecting certain cytokines. And just like the adaptive assays, immune assays, there's not sufficiently demonstrating the sensitivity or not providing sufficient details on the methodology can also be what we commonly see. And finally, realizing that there's a potential of a formulation effect. So the formulation of the drug product may affect the assay. And lastly, if you want to learn more or discuss more about these non-clinical immunogenicity assessments, we would like to invite you to this virtual workshop on this exact topic on January 26, 2021. The purpose here is to communicate the current regulatory thinking and considerations on non-clinical assays for comparative immunogenicity risk assessment for these generic peptide products. To get more information, register for the event, please check out the website at this link. With that, I would like to conclude my presentation by saying that the immunogenicity risks of the generic synthetic peptide product, especially those 5 listed in the guidance, need to be comparatively assessed to the recombinant RLD product, and this will include both the innate immune response and adaptive immune. The in silico assessment can enhance the overall immunogenicity risk assessment of these new impurities. So we recommend that you do that, as well as the in vitro assessment can also validate and verify your in silico assessment. Please remember, for both innate and also adaptive immune in vitro assays, the method justification validation need to be provided for your submission. With that, I'd like to acknowledge my colleagues and coworkers, and thank you for your attention. Oh, I forgot, there's also the challenge questions. Yes, so the challenge questions, number 1, if a proposed generic peptide product contains no new peptide-related impurities compared to the RLD, what non-clinical immunogenicity assessment assay is needed? A, in silico assessment on existing impurity. B, in vitro MHC binding assay on existing impurities. C, innate immune response on the whole drug product. Or D, no immunogenicity study is recommended. Answer is C. As we discussed before, the innate immune response on the whole drug product is still recommended, even though there's no new peptide-related impurity found. Question number 2, which of the following is not true? A, a proposed generic peptide product must use the same manufacturing process as RLDs. B, differences in manufacturing process could result in differences in impurities. And C, differences in impurities may affect safety and efficacy of a peptide product. Or D, comparative risk of immunogenicity in generic peptide product may be assessed through non-clinical assays. And the answer is A, a proposed generic peptide product must use the same manufacturing process as RLD. It doesn't have to be. As you can see, the generics can be synthetic and the RLD can be recombinant. with that, I would like to thank for your attention, and if you have any questions, we would like to take those in the discussion panel.